Introduction, Professor. Good morning, class. Today we will be diving into the fascinating world of molecular biology as it pertains to genetic disorders. Genetic diseases are caused by a multitude of factors, ranging from mutations in our DNA to alterations in gene expression. Understanding the underlying molecular mechanisms is crucial in diagnosing and treating these disorders. So, let's begin our exploration by delving into the different types of mutations that can lead to genetic disorders. Professor, frameshift mutations are genetic alterations that occur when nucleotides are inserted or deleted in a DNA sequence, thus disrupting the normal reading frame. This type of mutation can have severe consequences for protein translation, leading to changes in the amino acid sequence downstream of the mutation site. Frameshift mutations can result in non-functional or partially functional proteins, which may contribute to the development of genetic disorders. Professor, another important mechanism underlying genetic disorders is splicing aberrations. The process of splicing involves the removal of introns and joining of exons to generate a mature mRNA molecule. However, mutations can disrupt this splicing process, resulting in abnormal mRNA transcripts. These aberrant transcripts can produce non-functional proteins or alter gene regulation, leading to various genetic disorders. Professor, triplet repeat expansion disorders are caused by the expansion of repetitive DNA sequences, typically containing 3 nucleotides. This expansion phenomenon is observed in certain regions of the genome and can result in the abnormal elongation of a protein or a toxic gain-of-function effect. Examples of diseases associated with triplet repeat expansions include Huntington's disease and Fragile X syndrome. Professor, chromosomal abnormalities refer to changes in the structure or number of chromosomes. These alterations can occur due to errors during DNA replication, environmental factors, or chromosomal rearrangements. Examples of chromosomal abnormalities include Down syndrome, where individuals have an extra copy of chromosome 21, and Turner syndrome, which is characterized by the absence of one X chromosome in females. Professor, loss of function mutations occur when a gene becomes non-functional or produces a protein with reduced activity. These mutations can disrupt critical biological processes and lead to genetic disorders. For instance, cystic fibrosis is caused by a loss of function mutation in the CFTR gene, resulting in defective chloride ion transport across cell membranes. Professor, conversely, gain-of-function mutations involve the acquisition of new or enhanced protein functions. These mutations often result in abnormal protein activity or altered gene regulation, leading to the development of genetic disorders. An example of a gain-of-function mutation is seen in certain forms of familial hypercholesterolemia, where a mutated LDL receptor gene leads to increased cholesterol levels. Professor, epigenetic modifications refer to chemical changes that occur on DNA or histone proteins, influencing gene expression without altering the underlying DNA sequence. Disruptions in these epigenetic marks can contribute to the development of various genetic disorders. For instance, abnormal DNA methylation patterns are associated with diseases such as Beckwith-Wiedemann syndrome and Angelman syndrome. Professor, copy number variations, CNVs, are structural variations in the genome that involve duplications or deletions of large DNA segments. These variations can lead to changes in the gene dosage, affecting protein expression levels and contributing to genetic diseases. Conditions such as autism spectrum disorders and schizophrenia have been linked to CNVs. Professor, mitochondrial DNA mutations can also result in genetic disorders. Mitochondria are responsible for energy production in cells, and when mutations occur, respiratory chain dysfunction can ensue. This dysfunction affects organs with high energy demands, resulting in diseases such as Leigh syndrome and Leber hereditary optic neuropathy. Professor, non-coding RNAs, once thought to be unimportant, have gained recognition for their role in gene regulation. Dysregulation of non-coding RNAs, such as microRNAs and long non-coding RNAs, has been implicated in the pathogenesis of genetic disorders. Their aberrant expression can disrupt gene networks, leading to disease development. Professor, DNA repair deficiencies can result in a higher accumulation of mutations, increasing the risk of developing genetic diseases. Various repair pathways exist to correct damage, such as base excision repair, nucleotide excision repair, and mismatch repair. Genetic disorders associated with DNA repair deficiencies include xeroderma pigmentosum and Lynch syndrome. Professor, finally, genetic testing plays a vital role in diagnosing genetic disorders. Advances in sequencing technologies have enabled the identification of mutations and genetic variants involved in disease susceptibility. Combining genetic testing with clinical evaluation helps in accurate diagnosis, prognosis, and personalized treatment strategies. Professor, today, we have covered the diverse molecular mechanisms underlying genetic disorders. From frameshift mutations to epigenetic modifications and mitochondrial DNA mutations, each mechanism plays a unique role in disease development. Understanding these mechanisms is essential in developing targeted therapies and improving patient outcomes.